Please note that all attendees are in listen-only mode and that this webinar is being recorded. I'm Lori Sjogren, Community Program and Services Director for the National Ataxia Foundation, and I'll be assisting with today's webinar along with our Communications Manager, Stephanie Lucas. Questions for the webinar speakers can be typed in the Questions tab found in your control panel. We will answer as many questions regarding this topic as time allows prior to the end time of today's session. So, as your questions arise, please send those to us, and any unanswered or unrelated questions can be sent at naf@ataxia.org. Now I'd like to introduce NAF's Executive Director, Andrew Rosen, who will be starting us off today and facilitating today's webinar. Take it away, Andrew.
Thanks, Lori. Good morning, everyone. I probably, like you, am disappointed that behind me are not the Rocky Mountains, but instead NAF's beautiful office here in Minneapolis. It has been quite a week for all of us, but I do want to say thank you to you all for your remarkable support. This was as you probably can imagine, a difficult decision for the organization to make. We very much believe it was the right one, and I think even the news that has followed our decision this past weekend kind of proves that out. More and more organizations are doing the very same thing. But again, thank you for your remarkable support. The comments on Facebook, the emails, etc., have really boosted us all up in what was a difficult week. So we thank you for that. We are excited to have Dr. Jeremy Schmahmann join us this morning. Literally right now, he would have been walking up on stage in Denver. So instead, we thought we'd give him to you virtually, and he's going to be joining us here in just a moment from Boston. So again, thank you. We are working internally on how we can get even more of the great content that we had ready for you in Denver out to you all via webinars like this, and so stay tuned for more details about that in the coming days and weeks. And with that, I'd like to introduce Dr. Schmahmann. He is professor of neurology at Harvard Medical School and a neurologist at Mass General Hospital, where he is the founding director. In fact, I was there to help him celebrate the 25th anniversary of the Ataxia unit. And where he is also director of the Laboratory for Neuroanatomy and Cerebellar Neurobiology and a member of the Cognitive Behavioral Neurology unit. Boy, that is a mouthful. So without further ado, I will turn it over to Dr. Schmahmann. Thank you.
Thank you. Well, good morning. There you are. We're up and live. Well, thank you, Andrew. Good morning, everybody. This isn't how we planned on doing our talk together at the National Ataxia Foundation, but here we are, and ataxia patients and their families know how to roll with the punches and how to make the best of a situation that lands in your lap. First, I want to thank Andrew Rosen and Sue Hagen, Laurie Sjogren, Kelsey Trace, and Stephanie Lucas, all at NAF, who have helped manage the situation that we're in. And allow us to get to the point where we can sit and have a conversation today. So I'm not on the platform and looking at a large crowd of people, supporters, family, and so I'm going to make this a kind of fireside chat, just one-on-one, and share with you some of the ideas that I think are important in the current understanding of how we can go about making life better for the ataxia community and enabling you to be involved in drug development and the new therapies coming down the road. Let me start, if I can, just to give a sense of how we landed up this way. We had a medical research advisory board conversation late on Saturday night this last weekend, prompted by Stefan Pulst's conversation through email with Sue Hagen, who then forwarded that to some of us in the MRAB. And the purpose was to consider whether we should actually take a step back from having the meeting in person. And the conversation was very clear, and everybody shared their views and their ideas. And I think we were all erring on the side of caution that it's certainly the meeting is important in how we take us to a new place and develop drugs, but in terms of imminent necessity, that was not the case. And we decided to adopt a zero risk tolerance. And the only way to ensure zero risk tolerance was to actually not all be together in the same place. And as Andrew mentions, I think that the circumstances have borne out that that was the right decision to make. And thankfully, we have the technology now to allow us to talk in a one-on-one kind of a way like we're doing now. So just a word then about still on the coronavirus. Things are moving. The word that everybody's using is this is a fluid situation. We're learning new information daily, and the hope is to keep us all as safe as we can. The ataxia patient population has enough going on, and so being exposed to the virus would pose more of a risk than if you were completely healthy, and that was a major reason not to go ahead with the meeting. We know what we need to do. You need to wash our hands because our hands are basically traveling germ bags, is what we've understood now. The droplets of the virus can last on surfaces for quite some time, apparently up to a few days or a week, and therefore the transmission of the virus from a surface with the virus on it through our hands to our eyes, our nose, our mouth, these are things that can get us in trouble. And that's why it's so difficult to find Purell in the stores or Lysol or Clorox. But do what you can to keep things clean and safe. Wash your hands and try and avoid touching your face, which is such a natural thing to do anyway, that it certainly is a change of our own behaviors. And then keep abreast of what the Centers for Disease Control is telling us. The CDC is our authoritative site. Listen to your doctors, your medical providers, and let's all stay safe through this process. It will go away. A vaccine will be developed, and it'll be just like the flu in the end, where in our place you cannot come to work if you have not had a flu shot. And I guess the same will be like corona in the future, but in the meantime, we're just getting through it, and you and your families are as expert at getting through it as anybody else can possibly be. I think this is a good segue into what I want to talk about and what we have planned to talk about, which is your voice in drug and therapy development. And I think the point of this is, where we stand now, is that we are all patients. Everybody sitting in this conversation, everybody out there in the world is a patient. No matter how healthy we are, we have our dentists and we have our eye doctors and we have our GYN docs, and everybody is a patient somehow, somewhere. And I think that's an important place to start, because if you now throw our memories back to research in the mid-1900s, we know that there were some kinds of research programs that were engaged in that were, in retrospect, just egregious. And even at the time, I would think people were probably facing some ethical challenges about the way that they were conducting research on people as experimental subjects. That's where the idea that people have in mind still as a guinea pig comes about. I'm just a guinea pig in an experiment. And what I've just done with my face, you shouldn't be doing on a regular basis. I Purelled earlier, so just be careful of your face. We've become conscious of our own behaviors in a way we hadn't before. The change in research thinking prompted a whole array of institutional review board oversight, documents, committees, programs, requirements for investigators every year, no matter how many years we've been doing it, to go through those documents, read them again, and sign to affirm the fact that we've actually bought onto the institutional review board requirements for human studies research. So this is the intermediate stage, is being careful about providing informed consent, making sure that people who are taking part in clinical studies are doing it through their own volition, that it's their own agency driving their role in the clinical study. They understand the indications for the study, and now they're a subject in the study to investigate either a new idea or a science hypothesis or a new drug. And that's good and well. But we've now moved to a different stage, and I am so happy to see this, because what this stage is a recognition that, in a sense, that we're all patients, we are also all members of the research team. So the term that I have been struck by, not my term, I didn't invent this, it comes through the Patient-Reported Outcome Measures Institute, is recognizing that people who have a problem, and some kind of problem in medicine, are experts in their own disease. You are going through this, you know what you're experiencing, you know what matters to you, you know what the needs are that you want us to meet from our side of the table. It's a conversation always. And there are many members of the team. I'm going to emphasize that a few times as we go forward. And the point then is that you, as the experts by experience, are not research subjects. You are the research collaborators. We cannot find out new information about diseases unless the people who have those diseases are fully participating enthusiastically in a committed way and fully knowledgeable about what we're doing, so that we can get new information to provide new ways to understand, treat, and prevent the diseases going forwards. And that's what's driving the new approach, this idea that patients are experts by experience and that you are our research collaborators, which means that there's no hierarchy in the interaction in this kind of dialogue. We can't do this without you. You can't do it without us. We can't use drugs unless the pharmaceutical companies develop the drugs. We can't do this without the research coordinators, without the lab managers, without the institutional review boards. There are many moving parts to this, but the recognition and the point of our conversation today is that you matter and your opinions and your thoughts matter. So we've moved to a point of, in a sense, patient-focused drug development. And when I go through some websites with you in a little while, I'll show you how we can navigate some of the research websites that are relevant to you to make sure that you feel empowered yourselves to take part in this kind of a program. Now, all of this business of listening to patients and hearing your own thoughts about what's going on with you is really the key. That's how we get your voice in the development of new approaches to treatment. You know, we all have our areas of expertise. We're not expecting you to be biochemists and neurologists and imaging neuroscientists, but your willing participation and your knowing about the diseases and recognizing the importance of your role in this is key. At the same time, we need to listen to you. So listening to patients is something that not everybody's always as good as they could be at doing. And so listening, hearing, and then acting on what we've learned from you is essentially the approach that we need to emphasize and share with you and make sure that you know this is where we're going. And this is not just my own view. This is where the field has moved to in understanding patient feedback. So the kind of things that we're looking at would, for example, include a patient-reported outcome measure. Many of the folks at NAF completed the surveys that NAF sent out from my own lab to try and get a sense of what you are experiencing. And the work that we've done, just as one example, over the last year and a half, actually 2 years since NAF 2 years ago, is take all of your reported problems. We heard your thoughts, we heard your comments, and then we organized that into a questionnaire. And then we fed that back to people with ataxia in focus groups here. And then we fed that back to people around the US, and many of you took part in this, doing a retest reliability over a period of a month. And then we've been able to take that data set and put that together in a way that we have a patient-reported outcome measure for ataxia that will be useful, we think, and not only in clinical care, but in clinical trials going forwards. So that's one approach. But I think what I'd like to do is to actually tell you about one family's journey. And I think that this is so powerful because it gives us a sense of what is open to you at the present time now in real time to really move the field forward. And with the permission of the family, I want to tell you about the Martina family here in the Boston area. The family comes, as many of our patients in this area do, way back from the beautiful islands of the Azores. And they had this gene, Machado-Joseph disease, Spinocerebellar Ataxia type 3. that many people in NAF are dealing with. It's one of the most common, if not the most common, of the inherited dominant ataxias worldwide. This family, as you know, has a chance of 1 in 2 of every person being affected by the gene from the affected parent. And every time you spin the coin, you toss the coin, it's a 1 in 2 chance of getting the gene, the expansion of the gene or not. And it turned out that in this family, all 9 siblings got the gene. So now we have 9 people of a sibship, all of whom have the gene, and who have the chance of passing it on to each 1 or 2 of their kids. So what has this family done in the face of adversity? Well, firstly, they've recognized this. They haven't hidden it under the rug. They've talked about it. And hearing them, hearing Manny and Tony teasing each other about who's using a walker better. I mean, there's ways of dealing with adversity that you will. Alrighty. Let me pick up with where I was before. We were telling you about the Martina family and how they responded to adversity in the way that matters to us now, which is the approach to intervention to make life better by working with the research teams. So here's what I can tell you about this one family. We have the folks who in the family, firstly, they come and see the doctors. The reason that's important is it's important to share with other family members and with people who are afraid of the medical system or perhaps afraid of the diagnosis, that the old days of, I'm sorry you have ataxia, nothing we can do about it, it's a gene thing, you'll land up in a wheelchair, see you later, come back in a year if you'd like to. Those days are so over. And I want to emphasize that part of the challenge we have on the ataxia care side of this is to empower you, wait for it, to teach your doctors. So using the NAF website and the websites I'm going to share with you, you can get the information to share with your doctor. And your doctor or your nurse practitioner or whoever you're seeing will have the ability to know more about what it is that you're dealing with. And frankly, if your doctor or your caregiver doesn't want to hear from you about this, you should find another doctor, because we are all learning every day. The experts in ataxia around the world come to work every day and learn something new, because there are new genes coming out, there are new therapies coming out. It's a constantly evolving field. So if your doctor or caregiver doesn't want to learn something new, then truly find another doctor. So showing up to the doctor for working with physical therapy and with speech therapy and occupational therapy and taking vitamins that may be helpful, or taking medications for tremor or spasticity or bladder control. These are all things you can do actively, so it becomes your voice in the drug therapy of your problem. Rather than the whole field as an entity, it's about you and your care, taking care of you, because that's how you can help take care of your family. By being in as good a shape as you can then take it to your daily life. So that's the first part, is actually showing up, recognizing that there are things we can do, and no longer accepting from somebody who may not be an expert in the field that, oh, there's nothing to be done, because that is no longer the case. That's a sea change in how we treat people who have ataxia on a day-to-day basis in not only the ataxia centers, clinics, units around the country, but even the regular docs or neurologists who are in movement disorder centers or general neurology practices are learning every day. And our job is to actually share that information, which we're trying to do and will continue to do. So that's number one, the family shows up. And number two is there is open discussion about this. There is family planning. We now know about pre-implantation genetics. You can do things to help firstly understand the disease and move things along. But if you go beyond that, the family is taking part in the clinical trials. So Biohaven, I'll talk about in a moment, was first out of the box with a drug called troriluzole, which is a variation on riluzole for the treatment of ataxia. So members of the Martina family are in the clinical trial, and they're going to help us to understand whether in fact this drug works or not. We'll find out in time. And by teaching us on the care side how to do a clinical trial, that'll be important going forwards when other studies come down the road. They are taking part in the natural history study. what happens over time to the ataxias. That's a study that's being conducted across the country through the Clinical Research Consortium for the Spinocerebellar Ataxias. And then Jeanette's siblings, who are unaffected at this point, as far as we know, and hopefully will remain that way, are taking part in the ReadyScar trial. This is a study where we're looking to see when the disease actually begins. And the reason that's so important is we don't know when the gene starts to kick in to actually cause trouble. The reason that's important is when the magic medicines come down the road, which they will, we don't know when to start treatment. Do you start treatment when somebody has a problem with the balance, or with the speech changes, or they can no longer dance in high-heeled shoes, or they cannot skip anymore, or when they're age 15, no matter what? So we don't know that. Or when the MRI starts to change, or a fancy scan starts to change. This is all information that we cannot make up. We cannot just think about what it could be. The only way to get the information to feed back to you is to work with you and to explain to you why we're doing it, what we're doing, and make you recognize that you are honored, appreciated, and an equal member of the team, playing your part in the approach to get to where we're trying to get to, which is to improve the diseases, the ataxias, and therefore make life better for you. Jeanette has been a presence at the National Ataxia Foundation meetings. She's been part of the Birds of a Feather. She and the family and others have been part of the walk and roll locally to raise funds for NAF. They've helped raise funds for our lab, our own little group here, have come to our MGH Ataxia Center kickoff. She and other members of the family have been part of the new studies developing the prom ataxia, or working with us to find ways to measure ataxia more carefully and independently using the new tools of machine learning or wearable sensors, wearing an Apple Watch or sensors around the leg or the arm, to take this into a place where clinical trials can be conducted, not only at your visit into the hospital, but actually take something home with you. For example, the work we're doing now with Chris Gomez, also one of the members of the ataxia leading community, is working on a program to define the severity of disease through sensors and using the Apple Watch, for example, the Apple phone and the Apple Watch in people who have reduced ataxia. And there are a few sites around the country doing that. A number of our sites around the country are working together on brain imaging to see if we can find when the beginnings of the ataxia can be identified, not only from the genetics and the clinical phenomena, but from the imaging as well. So there are a whole range of ways that you can get involved and be proactive about ataxia. Raising awareness is what NAF's been so fabulous at. Raising funds to support the work is going to increasingly be important. And let me just diverge for a second into the Ice Bucket Challenge. The Ice Bucket Challenge that was transformative in ALS was not started by doctors. This was not a doctor-led initiative. It was not a research investigator-led initiative. It was the groundswell of people who were challenged by, moved by, and dealing with ALS. And that's where the fundraising comes from. You know, if you look at the amount of money spent in the political campaigns out there, the numbers are staggering. And you just wonder if we were able to marshal the support of patients, families, friends, and somehow get that $27, whatever it is around the country per person, you would be astonished at the good that can do. Being able to afford ataxia fellows, ataxia nurses, clinical research coordinators, all the things that are going on in the multidisciplinary ALS clinics around the country because of the Ice Bucket Challenge could be happening in the ataxia community because we need it. And as far as I can tell, the numbers are there are more ataxia people than ALS. So there's an example of being empowered not only in showing up with your doctor, taking part in clinical trials, taking part in natural history studies, but also helping to keep the momentum going from the patient side in fundraising, because that can come from the doctors. Each of our own centers raises small amounts of money for our efforts to continue. Grateful patients help us, and we're deeply grateful for that, because without that, we don't have the ability to do what we need to do. but the major donors, I think, are going to be coming through multiple numbers of people, and that's a part of your voice in the approach to the deeper understanding of and the improved therapy of these whole range of ataxias. This includes all the spinocerebellar ataxias, Friedreich's ataxia, multiple system atrophy. We have an MSA trial here as well. And so you're part of that team. So what I want to do at this point is, since I keep talking about teams, I want to introduce you to our team. Then after that, I'm going to take you through the website to show you how you can be proactive. So coming down, I want you to introduce a couple of folks in our lab. So this is Colin Valerian.
Hello.
And Brigitte Jacobi.
Hi.
And this is Jason Mcclure.
Hi, everybody.
And I think that many of you may actually have met Jason at NAF meetings, and you were going to meet Colin and Brigitte this time, but that didn't happen. So Jason, give us just a one-liner on Jason started off as a research coordinator and research assistant, and then lab manager, not a project manager, and he's the guy that makes stuff happen here. So give us a one-liner on what you do in the background for a clinical trial.
One line.
A couple of lines.
A couple of lines. So basically, I handle a lot of the regulatory reviews for things. I tend to try and attend all the investigators' meetings. I generally am listed as the lead coordinator, so then I get to go and train my other coordinators. So basically, I kind of have my hands on everything. I handle contracts, I handle budgets, startups,
And the
IRB, which is its own job all by itself.
Thank you. And Brigitte, what tell us about the project that you're working on?
Yeah, so I'm working on the project with multiple systems atrophy, the clinical trial happening with Biohaven. So I'm the person who you'll be contacting when you want to set up an appointment, learn more about the trial, and I'll be helping Dr. Schmahmann out running any visits that you attend, making sure all the paperwork is organized, completing the blood draw, and doing the sitting and standing and lying down blood pressures.
Terrific. And Colin?
I'm a very similar role, but I'm also working on a clinical trial for the spinocerebellar ataxias, mostly 1, 2, 3, 6, and 7, and also doing blood work, ECGs, paperwork, things like that. Brigitte and I are both here to help facilitate your transition into the process of clinical trials, and we're here to help along the whole way.
Awesome. So what I can tell you is that this is, the reason I wanted to show you our team is, this is a team sport. There are research coordinators around the country. The PIs I'm going to show you all work together with people who are actually doing the hands-on work. We do the exams, but checking you in, doing the lab data, doing the clinical rating scales, checking in with you, making appointments. This all relies on a team like this. And so the reason I want to show you our team is, you're on that team, and your role in that team is pivotal. None of us would be here doing this if you weren't part of the research program with us. So I think what I'm going to do is thank you guys for coming and joining me. And I'm going to now switch to, you may want to stick around in case I get myself all mixed up here. I'm going to switch now to screen and take you to some websites. So let me minimize this a bit. Am I still there? I'm still there. So let's go. So how do you get onto these websites? I'm going to take you.
The NAF research page closed.
So I'm going to take you to. So if you go to your website, you go to your computer and you type in ataxia.org. You have to spell it right. Ataxia.org. And you probably know this. Share my screen. Yeah, where's my screen? Yeah. People can see your screen. Okay, good. You can see my screen. Okay. So I typed in ataxia.org at the top, and it took me to the NAF website. So this is a resource that you can all use, and the reason I want to show this to you now is to take you to the research page. And on the research page, you can find the ataxia patient registry, clinical trials, research participation, a natural history study, and the tissue donation program. And just a word about each of these. The ataxia patient registry includes Friedreich's ataxia, Fragile X, and then all the different spinocerebellar ataxias. There are patient databases for rare diseases like RSACs, and also for the ataxia telangiectasia children's projects. This is how people know where you are and how to find you to enable you to be part of these clinical trials. So the next part down in the clinical trials of the ataxia.org, the NAP's website, will tell you about these different studies. One of them that is good to know about is a site called clinicaltrials.gov. It's a governmental website, and you can look up any kind of disease about trials going on anywhere in the NIH-related field. All right. You can go to the Clinical Research Consortium for the Spinocerebellar Ataxias, also on clinicaltrials.gov. It's run by our colleague, Dr. Ashizawa in Florida. Many of you are in this. Sorry, in Houston, Texas. Where did I say? Florida. No, he's in Houston. He moved to Houston. So he's in Houston. And this is the clinical observational study of spinocerebellar ataxia over time. The troriluzole study is through is Biohaven. It's listed on clinicaltrials.gov, but it's also the Biohaven itself, Biohaven Pharmaceuticals, has an SCAtrial.org page that tells you about the study. They need more people for this. They're looking for numbers of patients with SCAs 1, 2, 3, 6, 7, as well as 8 and 10, and they have now reopened for SCA3, which they hadn't done before. So if you're interested in being part of a clinical trial for troriluzole, that's where you should go. There is a study for Friedreich's ataxia. There's a study for multiple system atrophy that Brigitte just mentioned to you, also through Biohaven. And then there are, through research participation, there are other sites listed here that you could go through. I won't make you do that now. But you can look at these and see what is available. Here's a natural history study of SCA7, for example. You can find that one. There's the ReadyScar study I just mentioned to you. This is looking at people who are at risk of SCA1 and SCA3. In other words, somebody in your family, your mother, your father, your sibling actually has it. You either don't have it or you have really early symptoms. That's the study looking at where things can develop over time, and it's a really important study. The IDEA exchange study I mentioned to you earlier about the work we're doing with Chris Gomez and the APDM, looking at wearable sensors to see if we can figure out how best to evaluate ataxia in the real world space when you go home. And there are other approaches looking at wearable sensors, biomarkers in SCA3, and so on. And on the ataxia website, you will find the universities and the ataxia centers around the country that are particularly focused on the ataxias, and the links are now active as of yesterday. This is active and correct. For example, at our lab, you could click on Jason Mackmore's email. You just met him on the computer. Dr. Perlman has, Dr. Opal, Dr. Subramani. Everybody has a coordinators who work with them who can help you be involved in a clinical trial or studies that could be taking place at that site. Now, this is true for all the places you see listed here. Everybody is very invested in the ataxia field. If you don't have some place nearby you, be in touch with Sue Hagen and their team at NAF, and they can guide you to what's going on. There are also other studies, for example, the PROM study that we just completed, or the studies of other cognitive disorders in ataxia that we're looking at that may be available to you if you talk with Sue, and she can guide you if you cannot find it on the website. And then finally, and I'll end off here before opening for questions, is this concept of tissue donation. The brain donation is a key part of our care of people who have ataxia. So there's a fact sheet here about what brain donation is, the tissue donation for ataxia research. That's on the website. And there is a fillable form here that you could send in as well about the brain donation program. So I'm going to go back on camera and I'll stop showing the screen. And to make the point about brain donation, you know, they say that in life there's two certainties, you know, death and taxes, and we don't really like either of those. So since there isn't much you can do about the taxes, can't get out of that. And we certainly none of us is getting out of here alive in the end. The question is, in a disease or set of diseases like the ataxias, where these are rare disorders and we're still trying to figure it out, we can get so much information from the clinical exam. We can get information from imaging studies. We can get information from biomarkers. But in the end, the problem that's happening is the brain cells themselves get attacked by these gene disorders. And for us to be able to understand what's actually happening in the nervous system is a key piece of information about moving the field forward. So one can continue to participate in the research program even after passing. It becomes a gift to family, a sense of closure. This is certainly what the diagnosis was. We know, in fact, what it is. If there's a question in the future, was it really this? What was the nature of the involvement in your mother, your father, your uncle, your aunt? People know exactly what it shows. And it also provides a gift in perpetuity for the research field, because we can always go back in time to take the samples from someone who passed away years, even decades earlier, to ask new questions. And I can tell you there was work going to be presented at the AIM meeting now, just a few days ago, by colleagues here who are in the Alzheimer's research program, who are looking at the interaction of the gene for SCA1 and its risk for Alzheimer's disease. We know about the risk for SCA2 and ALS, and SCA17 looks very much like Huntington's disease. So at some point within the genetic interaction in these different diseases, we are able to use the patient material, the actual tissue from brain and spinal cord and peripheral nerves, to get deeper insights into what's going on and how to then prevent disease from taking place. So it's a piece of the puzzle that is critical to understanding everything we've done before in terms of the clinical exams, the biomarkers, the clinical trials. What do we know about the actual disease state? Which brain cells are working right? Which ones are not working right? What's the nature of the mechanism that's gone wrong in those cells? And can we find new ways to treat based on what we've learned about the brain from the brain donation program? So I think I'm going to stop at that point. And I hope I've given you some sense of how we value, honor, and appreciate your engagement as the experts by experience and our research collaborators as we deal with this field going forwards. From our side and the clinician and the caregiver side, we get up every day with the passion to make a difference to your life. You get up every day living the experience of what's happened to you. And together we, I think, have increasing opportunity with all the technology that's advancing and all the changes in the actual behind the scenes understanding of what we can do together. We'll get to a point in time where we are going to be better at treating, preventing, and even curing these diseases. And I know that together we can get there. And I thank you for your time and for listening. And we look forward, both myself and everybody around the country on the clinician caregiver research side, we look forward to working with you going forwards to enable you to have your voice heard in the drug and therapy developments coming down the road. So thank you very much. And if there are questions that are coming through, I'll be able to hopefully address those. So I'm going to make sure that Jason, who's here still, can help me do this. All right. Is there anybody out there who can help where we go from here? Okay. Is there any way to donate SCA affected embryos? That's a great question. That will be part of the brain donation program. Laura Ranham, who is part of the Medical Research Advisory Board, is heading up our little subgroup. And I think that's a question, it's a targeted question. If you would send that to Sue Hagen or the folks at NAF, we will probably be able to answer that more clearly. Are there any other questions on here? What do I do? Yeah. Okay, so all kinds of questions in here. All right. Is there a drug therapy or physical therapy available to lessen muscle rigidity of limbs? The answer is yes. So why do muscles and limbs get stiff? This is either spasticity or dystonia primarily, and there are medicines that work on both of those. The spasticity and the dystonia that it comes along with the ataxia sometimes can be very painful. Medications like baclofen have been around for a long time. Sometimes even muscle relaxants or the benzodiazepines can be helpful for this. Prevention of cramps with maximizing your metabolic milieu like magnesium and calcium and potassium can be important. And working with physical therapy can also be helpful to try and give you the ability to improve range of motion and decrease the discomfort that goes along with the spasms. The next one is, are there other diseases that cause ataxia from a smaller than average cerebellum? So there's, this is a good question because it gets at what these terms are about. The major words in this sentence are diseases, ataxia, and small cerebellum. So ataxia is impairment of gait, but the way we think about it is ataxia is a problem of gait that comes from cerebellar troubles. And the disease is something that attacks the cerebellum, whether it's a gene disorder or a stroke or a hemorrhage or an infection. So there are many things that can do that to you. And sometimes we can find the genes and sometimes we cannot. So as sophisticated as genetic testing has become, we don't always get the answer, even though we are going all the way through the repeat disorders like the spinocerebellar ataxias that we've talked about. But there are other ones that are rare that we can increasingly find with the sophisticated gene testing, but there are still some that we cannot yet find causes for, even if they run in families, and those, I suspect, down the road will be identified. One example of that is a disease called CANVAS, or cerebellar ataxia, neuropathy, and vestibular areflexia syndrome. The reason I'm mentioning that is it runs in families. It's a recessive disease. We couldn't find the cause, and the reason is that there was a strange gene disorder that was discovered last year that causes that. It was a particular clever way of looking at the genetics to find what the cause is, and before that we couldn't find it. So I suspect that'll be a big part of how we figure out what is going on here. Now, I see a note that we can, if people want to ask your questions verbally, you can do that too. So I'm going to, I guess, Laurie, if you can help with that, we can do that. I can look at another question here. On the news.
Absolutely. So if you would rather ask a question verbally, please raise your hand using the hand raise tool in your control panel. Looks like David Hubbard may have a question, so I'm going to unmute you, David. Let's see. Try to unmute, David.
Okay, yeah. Am I here? Can you hear me?
You're on. I can hear you.
Okay. Did you mention that SCA3 is most common hereditary?
I think that worldwide SCA3 and SCA1 are pretty close. Those are the most common, yeah, in terms of adult ataxias.
That's good to know then. Thank you.
Welcome. Yeah. So someone asked about CRISPR in a patient to treat blindness. The hope is that CRISPR will be helpful. This is an exciting new field. I'm not a geneticist. The geneticists are going to give us new insights into how we can get into the mechanism, actually open up the hood of the car and get in there and fix the problem. That's where they're going with this. It's very exciting. But I can tell you that if you go back to baseline, back to basics, the ability to figure out whether a gene is working, unless it's a miracle drug overnight and someone gets better within a few days, that kind of ability to detect improvement is going to require the clinical skill set that we are learning from the clinical trials. So yes, it's exciting. I don't know if we're there yet, but hopefully we'll get there. That and the other gene approaches, and there are many different kinds being evaluated now. But together with you and the clinical trial approach, we'll be able to figure out which of these works and perhaps which of those do not. But yes, that's an exciting development. So I see a quote here that I've been tested for all the genes and no hits were found. I still might have a defective gene, but researchers haven't found it yet. I'm still unspecified. What can those with unspecified ataxia do with regard to research that are specific to the patient? That's a good question as well. You guys are awesome. How do you get involved in an ataxia research program if they don't have a known gene? The studies that are designed to look at a specific gene would not be for you. But for example, the primary ataxia we just sent out, or the cognitive scales, or the physical therapy interventions, or imaging studies, all of those would be open to you because they're not gene specific. It's ataxia specific. And I suspect that if you stick with your doctors and let them keep looking at the genetics as time goes by, moving from exome to genome or more clever ways to figure out the gene, they will in the end get there if this is truly a hereditary disorder. And some of the recessive diseases with different kinds of genetic mechanisms are being unraveled all the time. So hang in there, all is not lost. I think they'll get there in the end. I'll take some more questions verbally. Go ahead.
All right, looks like Denise has a question. Denise, are you there?
I am here, yes. Thank you very much. I'm actually calling from North Carolina State University during my lunch hour, so hold on 1 second. So I have idiopathic cerebellar ataxia, and I was diagnosed officially last year. And I had the Athena panel done, and there was one gene though that came up with a result that was variant heterozygous missense, autosomal recessive, unknown clinical relevance. So I'm just wondering, like, is there any way we can actually put this information in somewhere so that if there is a discovery made, they say, hey, you know, Denise has this was diagnosed, because I don't think anything happened. I was just told by the neurologist, well, it's not genetic. We have no idea why 7 years ago you started showing this. It's idiopathic. But because there was something that did show up on that Athena test, I'm just wondering, where does it go? Is there a way for us to register this so that if something does show, we're alerted?
I think the question gets to a challenge that people are facing all over with this. And there's both a science challenge and a follow-up challenge. The science challenge is if you're finding a variant of unknown significance in a gene that is known to be a dominant gene that goes from generation to generation, you could only have one gene and it could potentially be relevant. If it's a recessive gene, then you're going to have two of those, one from each parent. So finding one copy of a gene that they're not sure about is probably not the cause of your ataxia. I haven't seen the specifics, but in general, you need to have two copies, one from each parent. And if they were both similarly a little different, that would be indication to keep going looking at that in more detail. But the single variant of unknown significance in a recessive gene would probably not be the cause. But there is one step further to go, maybe even two. Where we go from here, and so we've adopted a different approach to this. I learned this the hard way. The hospital came to me a few years ago and said, you know that when you send your drug, your gene testing to Athena, you cost us $1 million last year. And I said, did I really? So they weren't happy about that. And I realized that we need to do something different. And we have, and we've had a major impact on our cost, which is important to everybody. The way that we approach this is we look to see, is there something obvious about a person's clinical presentation that suggests a specific gene diagnosis? Sometimes there is. Someone looks very much like a Friedreich's ataxia case or a Machado-Joseph case. If that's the instance, then we can go for a small number of genes. But if we don't have that, we make sure that we've ruled out what's called the repeat disorders, because the way that the rest of the gene testing works, it cannot find those. You have to go looking for them specifically. And there are not that many of them. They're common in the ataxia community. SCA1, 2, 3, 6, 7, 8, 10, 17, DERPLA, Friedreich's ataxia, Fragile X. These are the common repeat disorders. If those are all negative, what we're doing in our group is we're going directly to exome analysis. And exome is going to transition to genome testing coming relatively soon. That's a much wider net to catch other genes. And so I would not rest thinking that you have a diagnosis based on this one heterozygous variant of unknown significance, but you can talk to your doctor about going further with exome and or genome testing. That's how I would approach that for you.
Okay, thank you very much.
Welcome. So I see here, should tissue donation be decided on ahead of time because of time-sensitive matter? Absolutely, and I'll tell you why. Again, this is from Cindy DeMint. The reason for this being so important, Cindy, is because it gets your family off the hook. They don't have to think about, would this person have wanted this? Are we honoring their wish? And it all goes back to the whole point of my time with you today, which is honoring you and making sure that you are being heard and you are being listened to. So if this is what you want, by filling out paperwork in advance saying, yeah, this is what I want, there's no decision to be made after somebody has passed. There's no agonizing. It's already a done deal. And often I will get a phone call within within minutes of somebody passing, because we've had the conversation before. The relationship, the trust that's evolved over time has gotten us to a point where we can say, this is what this person wants. Here's how we can go about doing this. Here's what happens at the time. And you can take care of the grieving process unaffected by all this other mechanics of what you do in terms of brain donation. So the answer is yes, it should be advanced and decided ahead of time for all those reasons.
Jeremy, it's Andrew here. I just want to add, right on the research page that you were on the NAF website, at the very bottom under the tissue donation program, we actually have a fillable form that allows us to collect information from you all ahead of time, so that when the moment does come, you're not scrambling and we've got all the information we need. So we would certainly encourage anyone who's interested to go ahead and fill out that form ahead of time.
We have a question here. Is any work being done on acquired ataxia? The answer is yes. I mean, there are many things that do this to you, and I can give you an example of people. The one that I'm fired up about is post-infectious cerebrolitis or immune ataxias. There are many causes of ataxia that are not genetic. Children often get an infection and then they get a little ataxic for a few days and it goes away. So the young kids who get a post-infectious ataxia is they get a bug, a virus of some kind, not coronavirus, but some other bug, cytomegalovirus, Epstein-Barr virus, and then the body reacts against the virus, but also gets its signals crossed and ends up attacking the cerebellum. In children, that generally goes away within days sometimes, but not always. And in adults, it can be devastating. So when we see this in adults, we can go in and treat that person with immune modulation, with intravenous immunoglobulin, and stop it cold. So you can save somebody's life by recognizing the immune attack. The same is true for some of the immune antibodies that we know are associated with either paraneoplastic, associated with a cancer, or other immune diseases that attack cerebellum. In terms of stroke and hemorrhage and other things that can affect the cerebellum, here we're dealing with this. The work goes on in the different disorders, trying to get at the underlying cause, but also this is where we're trying to find treatments to make ataxia better and treat the symptoms of ataxia and all that goes along with this, whether it's motor control or mental functioning, multitasking, mood changes, balance, dizziness, all those things that we can treat on a person-by-person basis. KCNA2 is one of the causes of ataxia, one of the gene things that can do that to you. So yes, there are different genes that affect the potassium channels, and that can produce different kinds of ataxia. So yes, you can get that. And I want you to take you to one more website. I'm going to put you back on my screen here. It says, are you seeing my screen?
Yes.
Okay, so let's go back. So here's the screen. I'm going to go to, in Google, I'm going to go, this is how you can teach your doctors. Gene reviews. Okay, one word. Gene reviews here is an NIH website. It is the definitive website. Gene reviews, and you can type in anything you want. So if you type in, so Spinocerebellar Ataxia, here's one, Spinocerebellar Ataxia, search gene reviews. And here's 6 and 14 and 11 and 10. So you can be the expert by experience with information in your hand. So here's one written by Dr. Ashizawa on SCA 10. I'll click on it. And here's a synopsis, and here's all the details of it. So I'll go back to me, I'll stop. So it's called Gene Reviews. All right, let me go back to the top of the page if I can. Gene Reviews. This empowers you to go where you need to go. All right, so I can stop showing the screen and go back to us. And that comes to the question of KCNA2. Can I comment on the quid pro quo for researchers to provide insight from studies, questionnaires back to patients and the broader ataxia community? To provide example with insight learned from a study has been shared. Are there resources for children of parents with ataxia regarding genetic testing? Oh, that's a different question. So I'm not sure what you mean by quid pro quo. I'll put my red hat on if you'd like. But here, I think the point of this is that the outcome of a research program is a publication. And this is why you can empower yourselves to go onto Gene Reviews or to Medline and find what's published, or just Google it and find what's been published. The information that comes out of the studies is being done in order to publicize for the other members of the research community and for patients to know what the results are. So yes, nothing's hidden, and it should be published. So information where insights learned from a study has been shared. This is what publication is about. So all the time, the entire literature comes out of research on patients shared with everybody. Are there resources for children of parents with ataxia regarding gene testing? So if your mother doesn't know her specific diagnosis but has pronounced symptoms, the place to start is with your mom. Because if you don't know what she has, then it would be difficult to know what to go testing for in you. I would talk with your mom about having a gene diagnosis. It's pretty straightforward to go ahead and get that now. The costs have come down exponentially. We have a genetic counselor who's part of our team who's been able to really make a big impact on interrupting the cycle of ever spiraling costs of gene testing. So that could be helpful for you to get your mom tested, then you can go from there. So we have a few questions from folks verbally, so please go ahead. And then Laurie or Andrew, just tell me when you think we should stop. You just tell me to stop and we'll go ahead and stop.
Sounds good. Looks like Kelsey's still self-muted, so we'll move on to another question here. Looks like Douglas, do you have a question?
Hello.
Hi.
So in my case, my genetic testing was fairly expensive, but we recently had a member of our support group who got a very low cost test done. I believe she is a patient of yours, but I was just curious what this involved because she got her testing done for under $100. I was wondering if you could comment on that.
Well, hi, Doug. Right, so we go back a while, and when we figured out what your gene was in you, this was a few years ago. And this
Right.
was before we had the capability to get a single gene analysis from a focused clinical exam. So in somebody like yourself, where there were a number of possibilities, you had to cast a fairly wide net initially. And I think in those days, we were using a company that was very expensive. I'm not going to go into, I won't mention names because that probably is not appropriate, but the company that we used was exceedingly expensive. We no longer use that company. But what was necessary for you was to cast a wide net to find the gene that was somewhat unexpected, but nevertheless explain what you have. There are circumstances where you can do a single gene test in somebody in whom you suspect the diagnosis, and that could be relatively inexpensive. The lady you're describing, I suspect, may have an insurance, and I think it may even be Medicare, that one of the companies is testing for us through the assistance of our genetic counselor, Helen Chen, who was able to find this. Being able to do this with an out-of-pocket cost for patients of $100. I don't quite know how that works. It's like magic, but she's able to do it. And that's part of the power of a genetic counselor who can look at the insurance, look at the different companies, look at the different policies to come up with a way. So there's a nuanced way to sort of dance around how to do this to come up with a testing approach that doesn't break the bank. So that's been in a state of flux, and we're actually in a good place now to be able to provide you with the powerful information that you need without really making it a financial hardship.
Thank you.
You're welcome.
All right, looks like Donald also has a question. Donald, are you there?
Yes, I'm here. I thank you for calling on me because I was beginning to despair. I did type my question above, or I can just restate it. And since we've already touched on the cost of genetic testing, that originally was my original question. How can we get a comprehensive genetic tax eval when Medicare does not cover it, and the company, and apparently it's a no-no to mention the name of the company, but I suspect it's the same one that was just being alluded to, quoted $99,999 for a private pay, and that's just a little bit out of my price range. So I do have, and this would be actually for my wife who's sitting here with me, a signed doctor's order from a Cleveland Clinic doctor who has already been through, and we've been dealing with this for quite some time, and she did feel like a comprehensive eval was necessary, since it was just touched upon about the prospect of getting a test for a single gene, but apparently we're way past that, but I still don't have an extra $99,999. So any suggestions with that?
I think the companies who are quoting you $99,999 should go to jail. Honestly, it's just.
Well, so do I, and a lot of people. And I was shocked when I found out that they're actually owned by a much larger, well-known national diagnostic company. I didn't even know that, but lo and behold. So one of the top two in the country owns this other one that quoted, and I'm not making this up, I'm not exaggerating. It was $99,999, and I just about fell out of my chair.
Well, you may want to send that little bill to your congressperson or senator or something. I just think that's completely outrageous.
Of course it is.
I think that, you know, there are ways to do this. I don't know the ins and outs, but our genetic counselor has found companies that are able to take Medicare and do the gene testing for an amount that reflects reality. What these people are basically got their head in the clouds. If you want to drop me a note, my email's all over the place. You can find me and see if we can help guide you. Maybe our genetic counselor can share some information with your doctor to see if they can help, because these are now attainable. It's really been a sea change in our ability to share information with you in a way that enables you, empowers you to take charge of your own life by not being stuck with no diagnosis, but knowing exactly what this is and what it means for you and the family and going forward. So this is part of therapy. It's like gene testing is part of therapy. We've talked about this in previous meetings, where this is called the need to know imperative. It empowers you. It takes the sting of not knowing away from this, even though it doesn't make the symptoms better. It takes away the anxiety and the uncertainty by giving you a name for this thing. And so there's a lot to be said for knowing what the gene is for all these different reasons, and it becomes part of clinical care. So if we can help at least direct you to the right place, just drop us a note and I'll see if we can help. But that's, I'm sorry to hear that. That's terrible.
Yeah. I hope she'll leave me unmuted for just a second. We're actually working with one of your colleagues that was on the list that you showed early on, and actually with two, one being Cleveland Clinic. I did not see that on your list, but also a university professor. And right now, nobody can figure it out. And in fact, the latest thing is we're told that she's an anomaly. We just don't know. And yet I've got this stymied situation. The Cleveland Clinic doctor.
Cricket has a question. Cricket, are you there?
Yes.
Hi.
And what is your question?
Okay. Can you hear me?
Yes.
Yeah.
Okay. Hi. Thank you. My husband Jerry has ataxia. Of course, we don't know what kind, but there was 10 people in his family that had the ALS, and has died of it. There's like 3 left. Some of them we don't think has true ALS because they've lasted a long time, but there are a few. But just recently, because of last year learning at the conference about the exome sequencing, we got that test done, and just last month they found a gene that tied, the 2 of his other relatives together with him with this KIF5A gene, and it's associated with ALS. But so we felt that was great to have something, just that being done. So thank you for mentioning that last year. So my question is, do you know anything about this gene, and where do we go from here? And does this mean that, my husband doesn't have true ALS, because he's had several tests done that said he didn't have true ALS. I mean, does that mean that? Because he had the muscle one, what was that called? The EMG twice, and it showed he didn't have that. That was a painful one. What do you know about this, and is there any trials? I'll go to those websites, and thank you, that was helpful. I'll go to those websites, that you gave us, and I'll punch in that gene.
That's right. So you can go right to Gene Reviews and type in KIF1A, kinesin family member 1A. It tells you it's a member of the kinesin family and functioned as an antegrade motor protein, et cetera, et cetera. So the data are all there. You are an expert you're a one click away from being an expert. So I'm not saying this to be flippant. This is actually real. You can go on the websites, and this is true for us. So this causes spastic paraplegia type 30, or sensory neuropathies, or maybe an ataxia, or things look like ALS. What's remarkable about ALS, and maybe that we can end off with this last point, is ALS gets all the love. It's a terrible disease. I feel awful for the people who have it. It's not something you want to have. But all the information out there in the world is about Parkinson's, Alzheimer's, and ALS. But what people don't know is that part of the fact that ataxia is more common than ALS, people who have the genes that cause ataxia have problems in exactly the same part of the nervous system that gets attacked by ALS. So SCA1 and 2, and sometimes 3, and some of the spastic paraplegias also have motor neuron damage that occurs as the primary problem in ALS. And so these are complex genes that produce complex impacts on the nervous system, focusing particularly on the cerebellum, but not exclusively so. So what you're seeing is exactly the overlap of why it's so important for us to keep the research going to increase the understanding of these disorders. I'll give you one more example. There's a disease called C9orf72, which has an overlap between ALS and frontotemporal dementia and cerebellar atrophy. So now here you have 2 major diseases, the dementia of the frontotemporal type and ALS and cerebellar disorders, all linked together through this new gene, C9orf72. And it becomes a virtuous cycle of research across the different disciplines that can provide new information. So I would, in your case, I would try and find through either websites or chat rooms or the ALS community, other people to talk with to be able to share your information. So not only can you be experts in your disease, but you can then become part of the research program that can try and figure out how best to help that. So become the expert, and then you'll print out these things on your gene and take it to your doctor and show them, whoever you're seeing. You go to the emergency room for something like a urinary tract infection, for example. Take your document. Here's what Dad has, or here's what my husband has.
Okay.
This is the problem that we're dealing with. So that you're teaching people about these rare diseases. The thing that's remarkable is that what we learned from the National Organization of Rare Disorders is that all these rare diseases themselves are rare, but together as a group, they're about 30% of neurological disorders. So you are the spokesperson, the expert, and the educator of people who don't know about these diseases. And you can do all that because you have a computer and a mouse.
Okay.
You can find the websites.
Okay. All right, thank you.
You're welcome. Thank you, everybody, for your attention. I see a lot of folks have stuck around through all of this. We only lost a few, so thank you. And thank you to NAF for the opportunity. Look forward to partnering with you along with all of my colleagues. I'm just a spokesman for all of our colleagues in the ataxia research community. There's a whole bunch of us. I got to be the lucky guy to spend time with you today, but there are many of us, both in the principal investigators, the research coordinators, the lab managers, everybody who works together, the nurses, the secretaries, everybody on our side working with you and your families as a team to get to a better place. So I wish you all well. Please stay healthy. Wash your face, wash your face, wash your hands. Don't touch your face, and be healthy through this period of time we're going through now with the coronavirus, because it will go away. And when we get the gene treatments for ataxia, so will ataxia.
And just as a reminder, again, I know Dr. Schmahmann pointed this out, but please consider joining the court's registry if you have not already done so. That is all we have time for today, but a recording of today's webinar will be made available, and additional questions can be sent to naf@ataxia.org. And thank you again to Dr. Schmahmann and his team and the NAF staff that contributed to this webinar. We truly appreciate all of you for joining today's webinar. We look forward to having you join us for future webinars, and take a moment to complete